I am here today with Dr. Timothy Garret. He's a professor at University of Florida. Before we get into that, we'll get into some introductions. I'm super excited about this show. For the next month, we're going to have scientists. We're going to have scientists on my show, and I'm super excited about that. I'll talk about why I think that's important. So today this is episode 10. I can't believe it. I started this during COVID-19, and a lot of I didn't plan it this way, but a lot of my topics have been on COVID-19, some about science, some about business, some about finance, some about the economy, some about constitutional law. And so today we're going to be talking about metabolomics in global health in relation to SARS-CoV-2 or COVID-19. Now, one of the things I was talking to Tim about earlier, and he so much agrees with me, and that's why I wanted him on my show, especially during these times where we're quarantined and we're isolated, and we're getting lots of information from various sources about science and health and so on and so forth. Tim and I are both scientists. He's an entrepreneur in academia. I'm an actual entrepreneur as well. I just happen to do it for a business and for profit. And so we'll talk about that in a minute. But how important it is for us scientists to communicate to the globe about what is going on in science. And sometimes that's about COVID-19. Sometimes it's about different things. And I think it's very important. How we typically have done that in the past is we write white papers. And Tim can probably follow up with me here, but you do some research, some experiments, you have a hypothesis, you do more experiments, you come up with a conclusion and so on and so forth. You do more experiments, then you write it up. This process takes months and years. Would you agree with that, Professor Garret?
100% agree.
Yeah. And the public does not want to wait months and years anymore because they are used to information immediately. And so that's where Tim and I can come in, and the Secrets of the Success podcast with me can come in. And our theme is never be outworked. And I want to tell you about Tim Garret, how I think that he has that mindset of never be outworked in a minute. So before we go further, follow me on Facebook, follow me on Instagram, on LinkedIn, visit my website at drsneedham.com. Follow me on my YouTube channel, Spotify, iTunes, SoundCloud, and you can call in today. We will be taking callers at 877-995-2111. That is on the event that I scheduled on Facebook and LinkedIn as well, and Instagram. So you can find the number there if you go check those events or my past posts. And you can find all previous, 9 episodes now. I think the 9th one was downloaded yesterday. All previous episodes on YouTube, my YouTube channel, Spotify, and iTunes and SoundCloud, and we'll update most of those. Most of those are on my website as well, drsneedham.com, but we also will have some of the ones later as well. So what we want to be is a voice of scientists to the global community, and that's what we're going to be talking about today. And again, social media is where it's at. It's immediate. People want that information immediately, not from a white paper that takes months or years to write and get to the public, or from even a conference. That's very important for us scientists to commingle with each other and network. But I think it's even more important to get our information out as soon as we can on social media. So without further ado, Professor Garret, thank you for joining me. I have always been impressed by all your work and what a great scientist you are. I'm going to just brag on you a little bit. He's not only a great scientist, but more importantly, he's a great human being, probably one of the best fathers and husbands and family guy I've ever seen. That he's just amazing. So thanks for being a great example for all of us, Tim. And I know your family's well, and you just have a great support group, and your family's great. So thank you very much. So, one of the things we always start out at is we always talk about the story of how we met. And sometimes that story your story might be different than my story, and we have not pre-planned this. So let's talk about that. But first, before we talk about that, describe your story. What's your story? What's your background? What do you think is important for people to know about you for today?
Oh, my background was one of adventure and one of sort of fortuitous events. And so I came into science, or came into college wanting to be an English student, and then took a science class and loved it, and fell in love with science as a sophomore in undergrad, and then started doing research in mass spectrometry as an undergrad. And so I had the great opportunity to play with instruments and to use a technique that was awarded the Nobel Prize later in MALDI, I didn't get the Nobel Prize, but I
So
used the technique.
So let's stop Professor Garret there just for a minute. He described a lot of words that we throw around all the time. Mass spectrometry, and I think it's important. Why don't you describe what mass spectrometry is to those that are watching that don't? You know how sometimes I describe it, but I want to let you describe it. You know, if you ever watch CSI, we use mass spectrometers for real and not just fake them like they do. So can you describe what a mass spectrometer is? Because we're going to be talking about metabolome, and I assume we'll be talking about that. What is a mass spectrometer for the general public, Tim?
A mass spectrometer essentially is a device, an instrument that measures the mass of molecules, or we have to make them ions. So we have to make them a charge to be able to measure them. But let's say tryptophan, a amino acid that's found in turkey, we measure the mass of that, and we can measure how much is present in turkey based on how much, based on the mass spec measurement. And so we can quantify what's there, and we can know it's tryptophan by its weight.
Awesome. That's a very it's what I like to say is it's a very, very high tech scale.
Yep.
To measure things, very high tech, but it's anyway. So we won't get into too much, but that's how you, and it's the most, I would say the most selective and versatile type of analytical instrument there is in the globe. Would you say that that's probably true, Tim?
It's 100% true, and it's used in every field, from forensics to environmental chemistry to food chemistry, everywhere.
Yeah. Everywhere. So that's great. And then you describe MALDI. That's just another term on how you make ions, because to control things in a vacuum, which a mass spectrometer is, you have to ionize them. And so you turn them into ions using different ionization techniques. That's all we're probably going to talk about for those. But I could go on to that for hours, and maybe some other day we'll have just a conversation on social media for just scientists in a group chat. So that's great, Tim. So, okay, that's awesome. That's a great story. You know, one thing that Professor Garret told me earlier, and I thought of how important this is, and if you talk to a lot of professors, and I know my professors were this way too, and he is a great group leader, by the way, at University of Florida. I've heard great things about him. And they want to impassionate people to join the sciences. And I mean, look at his story. English major, probably had a really good professor that, you know, was passionate about science, and all of a sudden he fell in love with it, and the rest of the course was his history. And so that's awesome. So you fell in love with it. You had English major, you fell in love with science. Keep going. What else happened?
Well, I graduated, and I actually got tired and went to industry for a little while, which is where I think my entrepreneur spirit sort of blossomed, because I worked at a company that was a startup, and I was working on technology that they're trying to, you know, take from the bottom up.
Yeah.
But I needed more. I knew I needed more work. And my dad, who was a PhD chemist and in polymer chemistry, he said, you have to really get a PhD to be able to make inroads in science. And so I took that to heart, and was lucky enough to be able to get into the University of Florida, and was lucky enough to work for Richard Yost here at the University of Florida. And then I studied more mass spectrometry and built instruments and looked at biochemistry, which is the study of how our bodies work and what molecules are needed for that. And then I, from there, continued on in professorship. And so I was lucky enough to get a professorship in the College of Medicine and then moved into pathology. And so this is where I am now, where I can conduct research here, and I can help other people do research by using the tools we have to help expand not just my group, but interact with other people around the world and the university.
Yeah, he, you know what? Tim's a humble guy. He's not telling you half of the things that he does, but I mean, he runs a clinical lab. He started a clinical lab. Well, now you're director of pathology. Tell us, expand on that, Tim. It's a great story. It really, really is, to think about where you've came from and what you've accomplished. And really, he has the theme of never be outworked, and that's the theme of this podcast. And it's just he embodies that mindset. So tell us more about what you do and what you've accomplished there.
Yeah, I mean, if you're never being outworked, that was basically been my motto. And not just that, but starting at 100% when you first go out the get-go, and keeping that momentum as fast as you can. Because I've always found that's what drives me, the work and pushing forward and trying to always be ahead of what might be out there, even though you don't know what might be out there, to be prepared to be ahead of that. And so I took over as director of experimental pathology as a way to help me learn more about administration, but also to expand in a role of learning more about the university and how I can impact the university through administrative aspects. And it's driven really by my passion for research here and my passion to make the University of Florida an excellent place as it already is. But you touched a little bit on the clinical part. Part of my role here has been to help our clinical lab use mass spectrometry in clinical diagnostics and help expand that role that our labs here at the University of Florida can do, and be able to do and measure more molecules and more metabolites in our body, to be able to help and improve health. And in the bottom line, that's really the passion, is being able to provide diagnostics that help improve health. That's really where I want to go.
So let's back up. He does an excellent job, and let's back up just a little bit. And you know, it's easy to get off track and say words that we think are normal to us. But he described a couple of things: the clinical lab and also pathology. And so he's involved in clinical labs, and he's also involved in pathology, using mass spectrometers and other devices to measure things in the human body. Can you describe a clinical lab and how that's important to the general public, and give us an example, Tim, and then also describe pathology as well and what's your role there and how that's important to the general public?
Yeah, that's very important for where we are right now, because diagnostic testing has sort of been forgotten about, or it's sort of not as exciting as surgery or other parts of medical world. But pathology is the study of disease, right? And not just that, but how we can measure disease and how we can measure sickness or how we can measure health by using blood or by using urine or by using other fluids that we have access to measure. And so a clinical lab is designed to use to collect blood, to measure metabolites, proteins, antibodies in your blood. Now those are all terms that we can talk about in just a second, but they're used to measure that, to basically help us understand how sick or how healthy you are. And 80% of all clinical decisions come through pathology. For whatever reason, whether that's a simple clinical blood test for glucose as a way to measure whether or not you may be diabetic or pre-diabetic. So glucose is one of those small molecules we can measure as an assessment of that. And it goes through a clinical lab. It might go through a quick analyzer first, but eventually is confirmed in a clinical lab where we can process and understand how that molecule relates to different diseases, and then send that back for a physician to make decisions on.
Wow, that's really cool. So before we get into talking about how important what diagnostics are and how he's measuring those, and how Professor Garret is measuring those, and then we'll talk about how it relates to COVID-19 and immunity and your health and so on and so forth, which I think is a very, very fascinating. We'll even get into some specifics that are fascinating to me as well. Before we do that, let's spend just a minute or two. Tell the story of how we met. Tim, I think I remember, but your story might be different than mine.
So from what I remember, we met for the first time in New Jersey at a meeting we used to attend called Clinical Pharmaceutical Solutions Through Analysis, through a colleague of ours, yeah. Mike Lee, who runs that meeting. And we met there sort of, from what I remember, as a way we were supposed to organize an event together. And
oh, that's right. Yes.
Organize an entrepreneur / a research program together to try to bring people in a room to talk about, how can science solve clinical problems? Sure. And how can not just science, but how can entrepreneurs and scientists together solve clinical problems?
That's all right. That's what I remember too. It was actually, sometimes those sessions could be kind of small, but at the same time, it was such fascinating discussions. And I remember that it was a great session. And I chaired that conference. And then several years later, you chaired the entire conference as well. And so great. And I like to talk about those stories, because I never thought we would be isolated and talking 3,000 miles apart, during COVID-19 on a podcast. And it's just know how important networking is. And I'm just very grateful for all the people I've met throughout my career, in my personal and my business relationships as well. And so thanks again, Tim, for being on. And so tell us, because we're going to be talking about metabolomics. Now, what I want to say about metabolomics is, and I'm sure even the general public has heard about some of these terms. Mm-hmm. There's genomics, there's proteomics, and there's metabolomics. I would say those are the three big ones. Would you say that's true?
Yep.
Okay. So this is just my opinion. I think the most important one is metabolomics. That's what Dr. Garrett is going to be talking about today. And the reason I think that is proteomics talks about, why don't you describe that and maybe why you think it's most important, and then I'll chime in.
Sure.
But why don't you describe the difference between proteomics, genomics, and metabolomics, and then I'll kind of add as necessary.
Right. So genomics is basically, that's what we're made of, genes, but those are genes that describe what could happen. They don't tell you what is going to happen or what will happen tomorrow. It just says you could have this susceptibility for this disorder, and that's important to know. But active disease is what we care about. And so proteins are of course translated from the genome, and they're the enzymes that turn over in our body. But they're much harder to measure in many ways, and they're much harder to know when they're activated or not. So we use something much smaller, and these are what we call the metabolites, the metabolomics, which are amino acids, or like tryptophan is an amino acid, and a very, very small molecule. The reason why they're important is because they're the reaction that happens. A small molecule goes in, and another molecule comes out of an enzyme, and we get the reaction rate of that specific product. So phenylalanine would be converted to tyrosine through a specific process, right? And so phenylalanine is one amino acid, tyrosine is another one. And we measure them because they're the true state of active disease happening instantly. So we know exactly that you have an immune response because the tryptophan and its metabolite kynurenine tells us about the immune system. And so we can understand that immediately what's happening right in your body, and use that as a way to say, yes, you are in an active state of disease, inflammation, or you have glucose, your glucose level is too high, and so you could be pre-diabetic or diabetic, depending on that. You may not have known that.
So that's what I love about the metabolome. And I remember it's been about 6 months ago now at a major meeting that Tim and I were both attending, and we were talking about some of his research and about the metabolome. And that's why, so I think proteomics are definitely important, and so are genomics. Genomics are our genes, what we're made of. And then there's proteomics, like Tim was describing. What I think is more important is we all pretty much, as the human species in general, have the same genome. It's very, very similar. But what we do to our bodies environmentally, do we smoke? Do we drink? Do we exercise? What do we ingest? That's going to determine our health. And that's why I think Dr. Garrett's work is more important, because it's measuring the health of what you do to your body. And it's real time. And so I remember having a conversation with him, 6 months ago or so at a major meeting. And it was about exercise. And I can't remember, he was thinking about having a grant. I can't remember what small molecule you were measuring, but about the health and what exercise does to you and how it's an antidepressant in itself. Do you remember that conversation, Professor Garret?
I do remember that conversation, and part of it was related to tryptophan, actually, because tryptophan is metabolized to serotonin, and serotonin is a neurotransmitter in our body, and it can cross the blood-brain barrier, and that's
describe what serotonin does for the non-scientist.
Yeah, so serotonin is a metabolite that makes you feel good, relaxed, and at peace, and that at least at the right level, it makes you feel comfortable and feels in an okay state, a relaxed state. And so a little bit more of serotonin might make you feel better, right? And so there's a connection between exercise, there's a connection between your microbiome, and that's another term, but basically the microbiome is what's inside your gut, all the bacteria that grows in there, healthy and unhealthy, and they produce metabolites. And tryptophan is one of these metabolites that's metabolized both by the body, but also by gut microbes that make certain products that are of certain metabolites that are then brought into the body through the colon and through the intestinal walls that are then transported into the blood. And tryptophan is one of those. There are bacteria that utilize tryptophan for food, and they spit out byproducts of that, which are then converted down into serotonin and other species that could be utilized in our body. So your gut and exercise has a relation to the health of your microbiome, and that could relate to how well you are producing or not producing serotonin if you don't have the right microbes. And so part of that research is understanding how and what kind of microbes might be present, but using a whole health system to measures your microbiome, measures your plasma levels of your blood levels of serotonin and your blood levels of tryptophan and other metabolites as well that can help us really make that work.
Well, that's fascinating. And what's fascinating to me is, and I just learned this a few years ago. I didn't know this, as scientists, we're always learning. And that's what I love. I want to be a lifetime learner till the day I die. Like you can tell Professor Garret wants to be. And serotonin is produced in our gut. I thought it was only produced in our CNS, in our brain system, but it's not. It's produced in our gut. And so what they're seeing now is what you ingest in your body can be highly affect your moods. And especially long-term, we talk about inflammation and cytokines that maybe Dr. Garret will talk about, but that can affect your moods. And so that's why what you eat or what you ingest or in your body and how you exercise, that's going to affect all your moods, all your chemicals in your body. And that's what I like about Professor Garret's work is he is actually trying to or actively measuring health or disease. And how can we get there? And what's the easiest way and, the most cost-effective way and the safest way, I mean, that's some of the things that he's researching. So it's very, very fascinating. And so tell us about the metabolome a little bit. What do you think would be the top 3 or 5 chemicals or molecules that you like to call them, that you would measure that the general public would be interested in? Maybe serotonin is one of them. And if you think you have to describe those. And, one of the most important ones is glucose because it's easy to measure. And maybe even describe why glucose is so important in the Krebs cycle and everything that we do. And then maybe 4 other chemicals to give people an example of what you're working on, which is really exciting.
Yeah. So glucose is obviously still one of the most important chemicals for brain chemistry. Without glucose, we wouldn't be able to survive because we wouldn't have the molecules that are produced from glucose that gives us energy and it gives us reactions that happen in our body. Too much glucose, though, causes other parts of our body to shut down and causes us to not metabolize or not produce enough of insulin or other ones to counteract the amount of glucose in our body. And so glucose is involved in hundreds of biochemical processes that go down in multiple pathways. But so eliminating glucose is not possible because it's an essential nutrient for our bodies to be successful. But controlling the amount of glucose, and glucose is a sugar. It's a single sugar, and you would get glucose from table sugar, which is a combination of both fructose and glucose, and your body breaks that down and produces both of them. And you might notice if you have too much sugar, you could get a stomach upset, or your brain actually can shut down a little bit if you have too much glucose, where you get a little foggy brain, or you get a little bit less. Your body can't handle that glucose load until it gets rid of it because there's too much in the body. So you still need glucose, but you don't need a high amount. That might be in a tablespoon of sugar. So it's still one of the most important ones. And I've talked about tryptophan, and I know that maybe too much a little bit, but I still think it's one of the most important molecules. It's an essential amino acid. So we don't make it. We have to get it from our diet. But it's involved in immune response. It's involved in neurotransmitter, not just serotonin, but also melatonin. And so that's the whole scenario. If you have a lot of turkey and wine, you produce a little bit more melatonin, you get sleepy. That is true. And if you have sleep disorders, it could be a mismatch between the amount of tryptophan and the amount that you're ingesting and the amount that gets converted to both serotonin and melatonin. And so knowing that conversion ratio could help in sleep disorders. It could also help in mood disorders. And so tryptophan to me is one, but the immune system part of it is important as well because tryptophan gets metabolized to another molecule called kynurenine. And there's a protein involved in that called IDO, or indolamine 2,3-dioxygenase, and that's just a name for the enzyme that process is taken into. But that ratio tells us how the activity of your immune system. And if your immune system is highly activated, you're moving tryptophan through the system, making more kynurenine, and it's working. If you suppress it, you're basically stopping the amount of tryptophan. And there might be reasons to suppress tryptophan. Say there could be reason in cancer. If you're in cancer therapy, you might want to stop your immune system to help another cancer drug be more effective. We actually use the ratio to understand organ transplantation. In infants, we have a couple of publications we published on taking both urine and blood samples, and we could tell these were monthly samples, and you could tell at which month the child was most likely to have an issue with rejection, or they would need more antibiotics by using the immune system activation. That was before they had a symptom or any other else. So their body started having an immune response before the rejection happened. And so measuring that could help you something. So that one, I think, and that ain't, so not just tryptophan, but other parts of the tryptophan pathway is very, very important.
So let's talk a little bit. Let's describe metabolite and metabolism. Describe that a little bit for the general public, and then I want to hear more about a priori testing of some of those infants that had organ transplants, because I think that's another segue we could go off into. So describe metabolism and metabolites for the general public that may not be scientists and know what that is.
Yeah. So metabolites are the small molecules that are in our body. And what I mean by small is they have a small mass. And that's why we measure them by mass spectrometry, because their mass is fairly small. You can measure proteins by mass spec too, and that they're just bigger.
But we make them into small molecules. Anyway, that's a whole other topic.
Another topic. We break them up and make it easier to measure. Right. But the mass of the metabolite helps us tell the difference between other ones. And a metabolite, think of your amino acids. So I talked about tryptophan, but all the amino acids that make up proteins, the individual amino acids are called metabolites. So taurine, it would be an amino acid, and that would be a small metabolite. Phenylalanine and tyrosine are both amino acids and metabolites. There are many other kinds of metabolites too. Bile acids, that would be part of how your body digests food and other things. But metabolism is the most important part of it, because metabolism is the turnover of those metabolites. So I use phenylalanine and tyrosine. Phenylalanine is converted to tyrosine, right, through an enzymatic process. That's metabolism. There are certain infants that have a disorder that prevents them from breaking down phenylalanine to tyrosine, and it's called PKU, phenylketonuria. And so when that metabolism is perturbed, they make too much, or they have too much phenylalanine building up in the body, because you can get phenylalanine from your diet. And that's a type of metabolism that can cause disease. And so that's one of them. That's a genetic disease that we can measure with metabolites, and that metabolism ratio of how much is being converted to one versus the other tells us really whether or not you have a metabolic disorder, and that metabolism is at fault.
Well, that's fascinating. And think about these essential amino acids that Tim just happens to be talking about. For example, tryptophan. And we ingest tryptophan maybe from a protein that we're eating. Let's just say turkey, beef, whatever. They have the essential amino acids, so we ingest those, and then our body changes it into other things. And I just learned today. I think he's told me serotonin before, that serotonin is basically a metabolite of tryptophan, but so is melatonin. Mm-hmm. And it just goes to show you how important what you eat is. And some of my podcasts have talked about the importance of eating, and you people that follow me on social media, nutrition is very, very important to me. And that's why I love Tim's work, is because it involves what the health actually is in your body based upon what you're ingesting and what you're metabolizing, so to speak. And that's fascinating. I want to get back into, so are you saying that you're based upon those ratios of tryptophan and another molecule, I don't remember the other one, in the infants? What was the molecule? Tryptophan and what else?
Kynurenine.
Okay, kynurenine. Are you saying a priori you can predict? That means you can predict before when they're going to have problems?
So we did in that publication that we published, and this was in the urine and blood. And we were lucky. It was a pediatric study in organ transplantation, kidney specific transplantation. And yes, when we collected all the samples, and so we didn't know it up front, and then we randomized all the samples. And that's something that scientists always do. And that's something important in clinical world to sort of tell you, is that when we do a research study, we want to blind ourselves to what kind of sample comes from where, so that we don't care where the sample came from, because in the end, we want to just use the data we get back to try to make what the right conclusions are for that, and what really is the right result, without knowing which sample came from where. And so we took these samples on a monthly basis from, I forget how many children were a part of the study now, but 12 samples over the course of a year. And you had normal trends where you could see they might have a seasonal flu, or they might have a cold, where they get a little blip, but it would be the successive increases that happen where you see next month it keeps getting higher and higher. And then you note that in the clinical notebook, they started receiving antibiotics to try to prevent, because they started to see a manifested problem. So we could see that early on, that yes, using the immune system and these two metabolites, and it really isn't, and that this is important sort of to note, it's not the quantitation of it, it's the ratio, the metabolism of tryptophan to kynurenine. That's
So when Tim says quantitation, he means it's not the absolute amount of it, it's the ratio of those two molecules that's important, correct?
That's exactly right. And that tells us that is metabolism. That's how much is being converted, because tryptophan we get by our diet. So you might eat more tryptophan in one day or another day, but the same amount is usually being converted to kynurenine or other products, unless you have an issue with the enzyme, or you have an issue with activation, where now you're making more of a substrate of a product, and that gets higher. And then the ratio tells us, oh, your immune system is activated. And so something is, the problem is it's not specific, right? So the reason we know these children probably had a transplant issue is because they're in a transplant study and they had a transplant. And we know that that's a factor of the immune system. But as I sort of said, they could also have an immune response based on the flu or even COVID-19, right? You could see whether or not you have an immune response based on an infection or a virus that you contracted, as a way to say, yes, you're sick in some way, but we need the clinical aspects to know that this is most likely related to organ transplantation, or this is most likely related to the seasonal flu.
Yeah, well, that's fascinating, because I think that's one of our jobs as scientists, and especially if we're in the medical community, is to try to treat disease, cure disease, not just the symptoms. So to be able to do that, you have to put together the right experiments to say, hey, this is what we should be doing before X, Y, and Z disaster happens. So Jessica's great work, and it leads into something else that we won't talk about now, but we'll get into later, that is fascinating to me, and I think it'll be fascinating to the general public too, as well. But so Tim, you've described the metabolome and what it is and all your work, and describe how is the metabolome a predictor of health, and describe that, gut metabolome, maybe what we call the microbiome versus other areas of the metabolome systemically, all over the body, or what other areas you measure. How is it a predictor of health? And talk about some of your recent research that you've found that the metabolism is a predictor of health.
Yeah, so we believe it's a predictor of health because it tells us exactly what's happening on a day in, day out basis. But it's really only a predictor if you measure over a course of period of time, and that say these metabolites keep showing up, and they're related to what's happening in your system, as well as knowing your exposome. You mentioned, Shane, early on, some of the metabolites that we're exposed to, those have a dramatic effect on the body, and we can measure that as a health and a predictive health. So if you're accumulating a metabolite from the air or from the water or wherever in your body, we can measure that, and we can say that this is potentially a predictive issue related to health. And glucose is an example of predictive health. We know that if you start to rise over a certain level of glucose, and you have maybe type 2 diabetes, and you don't start to take control of it or find a way to help revert that change in your metabolism, you're going to develop diabetes, and then you'll have much lasting effect. But you can predict, and you know that people are not, you can't predict it, but you know they're pre-diabetic and they have susceptibility to develop diabetes without taking control of that. The other thing is heart disease. There's some recent work in TMAO which is called trimethylamine oxide, which is a metabolite that appears to have a strong effect on heart disease. This was done, researchers elsewhere who worked on that, but it's related to your gut health, and it's related to what you ingest, and your body metabolizes this small molecule and makes it, and it appears to have an effect on your cardiovascular health, where higher levels of TMAO tend to predict those individuals who might have or develop heart disease. And but that's related to what you eat, and that's related to how your body metabolizes it. And so it's a key part of where we are. Some of the work in the microbiome that we've worked in is actually this oxalate. So oxalate is a small metabolite that causes kidney stones. And too much of it causes your body to obviously develop kidney stones, which are painful and have a dramatic health effect. And they're actually a microbe in your gut that is very capable of metabolizing oxalate out of the body. And it actually has been shown by our colleague here at the University of Florida to to actually pull out oxalate from the blood and take it through your intestine and get rid of it so that you can actually reduce your oxalate levels. But without that microbe in your gut, you don't have that ability to pull it out. And so some of the work we've been doing is to measure what metabolites allow that microbe to grow in your body, but what metabolites also might cause this secretion, because you may not be able to grow this microbe in your gut. And this is one of the issues with microbiology, is that sometimes even if you take a supplement, you may not be able to repopulate your gut with that microbe. But we know that there's a certain type of molecule that we believe is causing this excretion of oxalate. And we think that if we can give you that as a pill format, for those that have oxalate accumulation, we can start to pull out the oxalate that's in their body using a simple small molecule that would allow us to do that instead of repopulation, which may not work in some cases.
So that's fascinating. Did any of that work? Was it related to what they were ingesting? I mean, I've heard stories, and sometimes even in science, you can get conflicting papers and research and so on and so forth. And that oxalates can be caused, for example, by eating lots of broccoli. I mean, or is that true?
Yep.
And so can you just stay away from broccoli because it's not really an essential nutrient necessarily? So, or what did your work find there?
So there's definitely a long history of how many of certain kinds of foods that have high levels of oxalate. And yes, one of the key parts would be avoiding that if you have issues related to buildup of oxalate, and that would be a part. But but part of it is an inability of your body to metabolize it. So even avoiding those foods, you still might accumulate it over time, because oxalate is present in a lot of food, even at small levels. And so if for some reason your body cannot get rid of it, then you're going to accumulate it, and over time it might make issues related to kidney stone development more of an issue. So yes, avoiding certain foods, getting the right microbes, and maybe potentially getting the right therapeutic to help pull out that. And there's also a disorder that causes oxalate to build up in the body that is actually a liver disorder, and it causes a breakdown, and you can't get rid of oxalate in the body.
Interesting. Very interesting. So that's fascinating. And so you talk about how it can be a predictor of health, a metabolome. And so explain the importance of the prediction of, I guess, how the metabolome can predict immunity during COVID-19. And I know that you've talked about doing some research in this area because it's a very important area right now. A lot of academics are working on it. So explain how those are related, how that immunity could be measured, and how it would be important during COVID-19, and what your research would look like in that area.
So part of what I believe is one of the issues is actually related to lipids, and lipids are what you might call fat. Lipids are much more than just fat. They're molecules that make up your cell membrane. And so cells have a membrane, and that membrane has to be intact for them to be real cells. And so without lipids, you would obviously not be able to survive. But the reason I think that they're important is that some of the research has shown that when the virus, of course, when you're active with the virus, that virus has to penetrate your cells, and it has to take over your system to thrive. And part of that involves using some proteins that are spiked in that help the virus actually start the mechanism. But really what's needed is fuel, and we consider fuel food, right? So viruses still need food to thrive. And some of what we've seen in some published work that's recent is that the lipid levels that are present in your saliva or present elsewhere in your body tend to be part of that mechanism where this virus pulls in lipids to start replicating and growing, because it needs these lipids to become a full virus and full-fledged attack. And so there might be certain types of lipids that are more important for those that develop into COVID-19 versus a certain kind of lipids that for those who don't develop COVID-19 and may be asymptomatic carriers or not. We don't know, because obviously it takes a lot of work to a lot more samples, but that's part of what we're proposing, is using understanding what lipids might be present in your saliva that might help us understand how the virus could be used to grow and thrive, or not grow and thrive.
Wow, that is really, so why does the virus need lipids instead of, for example, glucose? Is it for fuel? Are they using lipids for fuel and then converting it into whatever mechanism for their own energy plant, or what?
Yeah. So one of the things that happens is that at least what some of the work has shown is it forms these membrane vesicles. So a vesicle is like a droplet. And so what it does is it creates this droplet around itself to protect itself from the outside world while it allows it to grow. So it's sort of hiding from your own immune system by creating these oil. You consider it like an oil droplet where the virus starts to continue to grow and then burst out and do more from there. And so that's sort of this kind of vesicle that you think if the virus is making that, then it's going to probably survive more. But if it can't make that, if it can't get those lipids to make that tiny, we call it kind of a microenvironment too. If it can't make that microenvironment, then it can't thrive, and it's just your body's gonna use its own immune system to find it and get rid of it.
Very interesting. Wow, that's cool. Well, good luck on that research, and definitely keep us posted. That sounds really, really good. So that's like are you currently working on that, or is that still in the hypothesis phase?
So we're not working on it just yet, but we have samples that we're starting to collect to measure those from and to measure that. And so it'll be a few weeks before we have some results to know. But it's not. There's other types of viruses that have something that does similar. SARS, the original outbreak of SARS, had some of this in it, but it wasn't really measured in detail with now the tools that we have available. And part of what happens in science is that, something that might have been studied early on by some techniques, they're foundational work, but now that we have new tools, we might be able to broaden the scope of what we can measure. And one thing I sort of didn't bring up with metabolomics is the essence of that we're measuring thousands of metabolites, not just 6. And you wanted 6 important ones, but part of what we want to do is measure many metabolites, or a huge number of them. And by measuring more metabolites, we believe that we can use informatics, and you might have heard of artificial intelligence or machine learning approaches, where we can use those kinds of approaches to help us predict types of disorders or what might be happening. And so by measuring many metabolites, we can make these associations. And lipids, too, are part of that. So they're small molecules, they're fats that are utilized. And so now we can measure 1,000 to 2,000 to 3,000 metabolites and lipids, and use all of that information to converge on a information, or you consider this sort of the new generation of information, because we have so many different metabolites that are present that we may not know what they all do right away. And that's frustrating in scientists, but when we discover something new, there's a lot of and we sort of forget this, is when you discover something and say you have a PhD and that's part of your dissertation, often you answer one question, but you open up 10 more questions because the research tells you that there's these 5 other avenues that could be happening, and let's study those too to help understand what might be happening. And that's what happens. And you sort of, the weird thing with COVID-19, you're seeing this live, where scientists publish a paper out very, very quickly, and then others might say, okay, well, I don't think that's happening, maybe this is happening. And so, and that happens in science normally, where I might publish a paper and then someone comes down a year later and publishes something that might be contrary to it or might confirm it. Yeah. And that's science builds on consensus. And when we have so much information coming in, all that information, some of it may be true, some of it may come out to not be true by other researchers studying it, and that's how science works. And so that's how we in metabolomics, by measuring lots of metabolites, we might figure out what they all do, but we don't necessarily know right away what they all do.
That is great to talk about. I mean, it is a great, I guess, summary of metabolomics and science, Tim, is that, and all of us, when we're deep into our dissertations, you described it, we're deep into our PhD research and we're thinking about these things, it is very difficult to stay focused because you're finding so many other things that you could explore. And that's where your research advisor has to say, no, just explore this one. The next grad student will explore the next one. And so
right.
That's what's cool about science. And, and that's what I think in our current situation, where I don't even like to say people are wrong or right. Okay. Because science is science and information is information. And I'm not saying it's totally wrong or totally right. It is what it is. And there are often 2 rights. There are often, because all you've got to look at the research. What tool did they use? What experiment did they use? Well, yeah, I can see why they got that why they obtained that information, and somebody might totally contradict it. But what was the term you used? Consensus. Science is consensus. What'd you say?
Sciences build consensus, right? And so research happens, you might have a hypothesis, but it takes more than one person to discover and prove that that works. And it might be that it works in some patients, but not others. So maybe it's possible it could only work in females, right? Or it could only work in males, or it could only work in a certain age group, depending on what happens. And so one study may have a really tight age group, or maybe really tight study, and that's okay. They might be studying something specific, but someone else may come in and add more subjects and see a different scenario happen. And that's true, especially with new drugs and drug therapies, where there is a different individual metabolism effect of a certain drug that may or may not be effective in one case versus another.
Yeah, well, that's wonderful. So, as we start talking about the future of measuring the metabolome, what do you think the future of measuring the metabolome looks like? And I'm going to have quite a few cool questions here, because this one fascinates me, like a lot of science does, but especially this one I've always been thinking about. So what does the future of measuring the metabolome look like in your opinion?
Yeah, I think the future is measuring metabolome on a regular basis. So like we measure glucose for those that have diabetes, you have to measure it every day. And that might be, well, not just every day, but multiple times during the day. And that might be frustrating at times. And we wouldn't want to necessarily measure every hour of the day, but I'd look at metabolomics as a way of, we need to measure it more often because your body's changing on a regular basis. And you have to set the stage to say, in certain years, your metabolism may change, or over the course of the year, your metabolism may be different. So I think measuring it more often is one of the most important things we can do.
And how would you think that would be? And I totally agree with you. And what that instrument looks like, I don't know. I'm thinking it's ion mobility mass spectrometry or something like that. A mass spectrometer, the problem with a mass spectrometer is you need a vacuum. And unless you do that in space, that's going to be really, really expensive or, powerful. So I don't know what that device looks like. What would that device do you think would look like if you were going to be doing it personalized?
I think it's going to be a way in which we can have at-home kits prepared. And you can send those samples into a normal lab where they would process those samples until we get to where you could have a small sample instrument or a small localized regional instrument. Or maybe the future would hold where we have, say, 300 metabolites that are measured in a very simple array. And you could have a small set of a small drop or whatever that measures those 300 metabolites to give you some kind of sense what's happening. But in the beginning, I think it's going to be at-home kits that we send out, and those come in to the clinical lab, and they're done in a very high clinical setting and done with traditional mass spectrometry approaches to try to build that part in. And there's some evidence that works really well. Obviously with the organ transplantation work that we did, but with nutrition is something that has to be measured on a regular basis to help to understand what might be happening with certain kinds of foods that you're measuring, or with athletic performance, where you're maybe something has changed in your body and you need to measure that metabolism as to why you're slowing down, or why the muscles aren't twitching at the right rate, or those kinds of athletic performance issues are important. And you have to measure on a regular basis to know that. And if you get hurt, you want to measure the recovery process, and what nutrients you need for recovery might be different than what nutrients you need for the next phase of at home or physical therapy, right? Depending on what kind of disease. And so I don't know if we really know that paradigm in terms of how we recover as individuals when we get sick, or what kind of nutrients you might need, or what kind of therapies you might need at different stages of recovery.
Well, I didn't have to ask the question, but this was what I was going to ask about is, for you, viewers out there and listeners out there that are thinking about this, and what does the future of measuring the metabolome look like? And this is already happening, and it is $1 million business, is major sports teams, whether it be a major league football team, soccer team, I should say, in Europe, or major league baseball team in the United States, NFL teams, they are measuring metabolomes constantly of their athletes. And let's just think about why that would be. So, like a pitcher in baseball, they have a pitching rotation, right? Because they think they have to rest X amount of days. Well, if you can measure their metabolome, you can predict how fast, like Professor Garrett was saying, how fast they will recover. So when they can pitch again, it is a big,
Yeah.
big business. And it's they're measuring them all the time. And
Yep.
so it's just you used to just be like, oh, I feel well. And then all of a sudden you hit a wall. Oh, I don't feel well. Well, can you predict when you wouldn't feel well? And I think that's some of your work that you're doing is a huge going to be part of the future and just in great, and not only probably in sports, obviously because athletic performance, but maybe even in a workplace to know what's optimal for the individual. If you can do all these things easily, test 300 metabolites easily in a personalized basis, you could even know for yourself what's optimal for you. So
I agree. And
it's great work.
And we already know it's less expensive to measure the metabolome than it is to say proteins, because newborn screening is, I always tell students that we have actually your metabolism has been measured the first day you were born. And
see, they actually talk about newborn screening. A lot of people might not know that.
Newborn screening is what we use in when every infant is born in the United States or elsewhere too. A small heel prick is taken, and small as a few drops of blood are put onto a card, and that card is sent to a clinical lab, and that card measures metabolites, amino acids, as one of the set of metabolites it measures as a way to know what kind of genetic disorders might be present, because the infant can't tell you if they're sick. They can't. They have no speech, obviously, and parents and the infant may not be showing any signs of sickness until the accumulation of the metabolite happens. And that's the example of phenylalanine and tyrosine, is that phenylalanine builds up. And if too much builds up, then you have brain damage, and then you have other issues. And the treatment for that is so simple. You just eliminate the amount of phenylalanine in the diet, and the infant will grow to be a normal, healthy infant. And that's why newborn screening works phenomenally well. And it's inexpensive. And we test over 4 million babies a year on it. And so there's precedent for using metabolomics in a way that for human health. Now, doing that on a scale in adults hasn't been done. It's doing in small areas right now, but not on a very US wide approach.
Yeah, and most of those newborn screening, I mean, I remember, having 4 children myself, all they tell you, you don't really get the results or anything. They just if there's a problem, they'll tell you. So right. if you never heard about it, you might think, well, my child was never had a heel prick and blood drawn. No, they probably did, but you just luckily didn't have any issues, and so it was a healthy baby. And so I think that's it's a great success story of newborn screening, of just how the whole metabolome for a general population, not just infants, could work. And just to tell my own story, several years ago, about a year ago, probably, I ordered my own glucose meter and my own ketone meter, and I was just going to test myself throughout the day. If I ate this, what would happen? When I exercised, what would happen? Now, why did I choose glucose and ketones? Because you can easily buy a kit and some strips from Walmart or other places, Amazon, whatever it is, and it's easy to measure. So you just got to prick your finger, get some blood, and it tells you. Mm-hmm. And it actually changed some things that I ate, changed how I did things, changed when my blood glucose was low, what I would do during that time of the day. And so it can make you feel a lot better. It's a great tool. That's an example of using your own metabolome to see what is happening real time in the body. And I can't wait for more tools to be available. Unfortunately, as a scientist, I always want to test things. So it could be crazy, right?
It could be. Yes, I agree.
Yeah. So this has been wonderful, Tim. So is there anything else you want to add to the metabolome before I get into my last question, which I think is one of the most important questions on my show? Is there anything you want to add to the metabolome, or tell the listeners about science, or something you're passionate about?
I mean, basically what I want to say is science has a huge role for where we are right now in COVID testing, but also where we are in the future. And diagnostics is what we're in right now, has a potential to really, really make our lives better when we do it in the right way, and when you have the right kinds of studies that come in. And so I encourage people to study science and look at places where you can get science literature. Now, it might be hard to read science literature at times, but once you start reading a few parts of it, you sort of can see sort of how science is built. And the strange thing on this virus is that there's if you go onto PubMed, which is a website in which we use to search articles, there's about 4,000 articles that have already been published on SARS, CoV-2. Not all of them have been peer reviewed yet because they're on these archive websites or whatever early, but that's a huge number of papers that have been published just on a specific topic. And that just goes to the fact that we're doing a lot of research actively, and that's all stuff from 2020 this year. And so it's a lot that happens. Yeah.
It's you know what, and it's great to be a part of that, about that part of that industry and a part of that science camp to be doing some of that work. And good job to you, Tim. Very, very good. So as any of you that have been following me for a while now or have seen some of my podcasts, is I always wrap up with an example of, we talk about failures in life. And I had a great guest last week where she said she didn't think there were anything such thing as a failure. There's just learning experiences and you learn from it. But everybody probably externally, I know in my own life, I'm sure people look up on my life and say, oh, he failed at that. Or I look at my own life and say, I failed at something. But what I know is that, that's not always the case. That I think there's only a failure if you fail to get back up and you didn't learn something from it. And so, Tim, why don't you give us an example of maybe, quote unquote, an apparent failure that turned into a success for you? And I could already think of some from your story, but I want to hear what you think it actually is. And so go, you're up.
Yeah, I agree that, if you focus on failure as the idea, you sort of lose sight in the learning experience that happens, right? And so it's really what I look at it as a unique learning experience. I have one from grad school that's sort of unusual in that I was spending a lot of time trying to fix an instrument, and I was, spent a great deal of time putting one together and trying to make it work. And then I foolishly, made a mistake and bumped it or moved it around, and it went off track. And that was about 17 hours after I'd already spent about all that time working on it. And so at first iteration was, I'm going to give up. That's it. I can't complete this. But what it allowed me to do is sort of refocus the idea and take a step back and rethink how I was putting it together. And I put it together in a much faster way the second time, and a better way that was foolproof and that enabled the instrument to work in a much better way. And so that's a simple example from grad school in terms of taking something that looked like something that might go wrong to something that should be better. And then in the end, we published the papers and developed an instrument from that that would then be commercialized. And so that's the kind of ideas that sort of, if I had let that one part of grad school stop me, I would have moved on and not made it further on.
That's awesome. That is just a wonderful story, Tim. And you've been a great guest. Thank you very, very much for your time. I know it's very precious to you. And so we're going to be wrapping this up. It's going to be next week. It'll be on Spotify. It'll be on iTunes. It'll be on YouTube. It'll also be on SoundCloud. And Tim, you've been a wonderful guest. Thank you very much. Thank you for sharing your story. Thank you for sharing about science. I usually take a 1-minute clip from the show to kind of highlight it, that'll kind of, tease viewers to go watch it once we've recorded everything and edited everything. I've made enough notes for about 4 clips. So thank you very, very much, Professor Garret. And you've been just a great, great guest. So we might have a caller on the line. Let me text. I think somebody might be trying to call. One sec. Can you take a call?
Sure.
Let's see. So 1 second here. I'm being my own producer today. Let me just verify that they're calling. So but you've been a great guest. And so
thank you.
While I see their response here, because I got to put them on hold and put them in a queue before I actually put them on live. I want to see if they really are trying to call the show or not. But how's Florida? How's the weather? How's everything there?
Weather is good. It's starting to warm up as normal, and we've enjoyed the time at home with family. That's one of the nice things, is that working from home allows us to sort of be with family and try to do stuff and be involved with, different things. So I can take a break and I can go outside and play with the kids for 30, 40 minutes and then come back and do work, right? Which is a really nice, wonderful thing. You can look at being home as sort of frustration, or you can look at it as, let me try to change up the scenario and let's see what we can do with what we have.
What a great mindset. Great mindset. So the caller didn't have any major issues or questions, so they were already answered. That's good to hear. Tim, you were a great guest. Hey, I'm gonna have you when we hear more about your research. I want to have you back on the show. You're a great guest. We're getting some great comments. So thank you very, very much, Tim. And I'm sure we'll see you around and be in touch.
Thank you very much for the opportunity.
All right. Have a good day. Take care.